Hello everyone. In this video I'm going to talk about chi-square test, and specifically one of the usages of the chi-square test, which is used for association. That's why it is called testing for association between species. First of all, let's see, what does that mean? What is the meaning of association? By giving some examples, I think you will understand what does that mean. A positive association. What do you think the relationship or the association between a predator and prey? Why do we say it is a positive association? Well, because they are always together, because one of them is looking for the other one. So this means that they are always associated together because one of them are hunting the other one. So they are associated together. The other one is like a herbivore and plant. So this herbivore needs plant to eat. So in an ecosystem, you will find them together because the herbivore for survival is always looking for a plant. So if these herbivore and plant are associated together or not. This is which is yes, and this is the meaning of positive association. Or another example, which is symbiosis. While they are giving benefits from living together, having benefits from living together, which is called symbiosis. So they are always together, so that's why they have a positive association. What is the negative association? Well, there are competitions that they are looking for different nutrients. So always they are avoiding from each other. They are not locating at the same area because they are looking for nutrients with a competition that they exclude each other. So competitive exclusion. That's the meaning of type of relationship that they have. Remember that if they are locating in a different niche, so because they are looking for different nutrients, so their habitats are different. So always it is a negative association. They are not associated together because they are looking for different things. But what about the no association? No association means frequently apart from each other. So there is no rule. You might find them sometimes together, sometimes not. And randomly, they might be seen in the same area, but there is no association between them. They don't have any relationships in terms of nutrients or other things. They just might be randomly at some areas, but there is no association between them. Okay, how do we calculate and say mathematically or statistically if there is association or not? Well, the answer is here by doing the chi-square test. The chi-square test will give you the answer if these two are associated or not. Before going to the example which is related to the chi-square test, we need to know some methods which will be used in this test. The first method is quadrat sampling. What is quadrat? Quadrat is a square. You can see it here. This is a square, and which is used in a number of ways. It will be used in estimation of population density. It will be used in measuring the distribution of a species, and it can be used by placing repeatedly in a sample area to provide a reliable estimate. So one of the reasons that you are using quadrat, you can find the number of the plants and everything in that 1 square meter. If it is a 1 quadrat square meter, then this means that here is 1, 1, 1. So that's why we call it 1 square meter. So this is the way that we are taking the information by finding or by measuring or by counting the number of the plants, the quality and the quantity. What type of plants are locating in this area of the quadrat and how many of them. So you need to calculate and count everything exactly. Here is the video. You need to watch the video by yourself. It shows an example related to a chi-square and the meaning of chi-square test. Okay, hope you have watched that video, and if you watch that video you will understand the meaning of the chi-square test. But here we have another example for chi-square test, which is the test of association. Here we have two different plants, marsh bedstraw and bottle sedge. These two plants we want to find if they have the association or not. How do we know that if they have the association? By using the quadrat sampling and by usage of the chi-square technique. Again, how? Okay, we have 100 quadrats that these 100 quadrat sampling we counted the number of the plants, the number of the quadrats in which the plants were together or were not together. So, for example, here, from these 100 quadrats, 12 quadrats contain both bottle sedge and marsh bedstraw. So 12 contain both, and then 3 quadrats contained only marsh bedstraw, 3 only marsh, and 29 contain both. 29 contain only bottle sedge. So 12 contain both, 3 contain only marsh bedstraw, and 29 contain only bottle sedge. So this is only one of them, this is only one of them, and this is both. This is how we count it from the quadrats. And the remaining quadrats, if you minus from 100, then the remaining quadrat will be 56, and 56 none of them. So 56 none of them none of the quadrats included neither this one nor that one. So how do we start calculation? We make a table. In this table, we know that the total number is 100 quadrats, and then we make the table according to this table. The presence and absence, for example, in case of bottle sedge and in case of marsh bedstraw, and we fill the table. Yes, once we fill the table, then you will see that, for example, in marsh bedstraw, we found 12 quadrats, both present. So 12 quadrats, both marsh and bottle sedge were present. And then in 29 quadrats, marsh was absent, but bottle sedge was present. And in 3 quadrats, marsh was present, but bottle was absent. And in 56, you will see that absent. Okay, so this is the way that you fill the table. And the next slide showing that, okay, the total number for each row and column, you fill all of them. How do you calculate the expected? This was the observed. In this table, you saw the observed value, but we need to know what is the expected value. How do we calculate the expected value? The expected value is so simple, just by like this, 41 times 15, okay, divide by 100. So row total times column total divided by grand total, which is here. So here would be 41 times 15 divided by 100. And then the same for here, same for here. So this will be showing the expected value. After the calculation, you will see how the expected value was, you can see over here. And then the next step is how do you calculate the chi-square? The chi-square formula, as you saw in the video, it's sum of this is the observed minus expected squared divided by, again, expected. So how do you do that? Let's see over here. This is the observed. So observed for each plant. So for each plant, observed minus expected, observed minus expected squared divided by expected, which is again 6.15 plus, and again in the next one, observed minus expected squared and divided by expected, and again the next one minus observed minus expected, again observed minus expected. So you will do for all of them, for all the plants, until you finish the number of the plants based on the observed and expected. How many plants did you observe minus expected and then divide by expected? Observed minus expected and divide by the expected. According to this, you will get four types of numbers that you will need to add all of them, and finally you reach to this one, which is the final chi-square. What happens when you have the final chi-square? You need another table which is called table of freedom. The table, that table needs to be known. What is the freedom? What is the degrees of freedom? Degrees of freedom, once you make that this contingency table, the rows minus 1 and column minus 1 will give you the degree of freedom. So degree of freedom of this would be 1, which means that here, this is degree of freedom 1. You just need to look at over this table. And once you're looking over this table, in biology, it is sufficient if we look at the 95% certainty level. So p-value or certainty level, if we say p-value as 0.5, which means that at the certainty of the 95%. So at the certainty of 95%, the critical value is 3.8. What is our critical value is 11.10. So our critical value, our chi-square, is higher than 3.8. This means that when our chi-square is higher than the critical value, which means that the association is meaningful, significantly higher than what was the least or what was the critical value. So our number is higher than critical value. It means that it is meaningful, the experiment is meaningful, and the association is positive. So we say that, these two plants are associated together. There is some meaning for these plants that they are staying together, because we saw it for many different quadrats that they were together. And this is the statistical analysis to prove that, yes, there should be some meaning from being together. Okay, this is an example of how you answer the question in your exam. Multiple choice. A study was carried out to determine if there is an association between two plant species that grow on a mountain slope. Why is it important to use a random sampling method? Pause the video and answer the question. Yes, that's right. To ensure that all members of the community have an equal chance of being selected in the sample. Next question. This is really nice question. The study was carried out to determine if there is an association between two plant species that grow on a mountain slope. A chi-square test was carried out and the data collected. The calculated chi-square value was lower than. What does that mean if the calculated chi-square is lower than? Yes, you answered correctly. This means that there is no association between the two species of the plants growing in the same habitat. Now, we already discussed about ecology, the study of the relationship between living organisms and between organisms and environment. So, if the environment is included, then that would become ecosystem, which is a combination of community plus abiotic environment, and population, which is just living organisms without abiotic factors. And then within the population, there are species that make that population. And if you have more than one species or more than one population, so two population makes a community. And community plus abiotic becomes ecosystem. So we have discussed this before. Let's move to the next slide, which is related to nutrient cycling. Nutrient cycling means the nutrients will be recycled because the supply of nutrients is limited. So the ecosystems constantly recycle the nutrients between the organisms. And how does it happen? It starts from producers or autotrophs that they convert the nutrients from inorganic form to organic form, such as they take the carbon dioxide by uses the energy taken from the light, makes the glucose, and then the heterotrophs that they are ingesting other organisms to gain the energy from them. And then we have saprotrophs that they are breaking down the organic nutrients such as bacteria and fungi and digest them outside. So that can be used again. And this cycle is important. So we can see the plant here and then decomposers, which are saprotrophs. They break down the molecules and then it will be used stored in the soil and will be used by the plant, and again plant make the glucose, and again this cycling will continue, go on and on. Ecosystems are sustainable, and how can we show this sustainability of the ecosystem? By making this mesocosm. This is a brilliant idea by this guy that he made this mesocosm in 50 years, and it is still alive for 53 years. I'm not sure he created this. If you just Google, this picture is very famous, and he created this mesocosm in 1973, almost at my age. Anyway, so to remain sustainable, an ecosystem requires what? It requires continuous energy availability. So a sustainable ecosystem needs lights from taken from the sun. A sustainable ecosystem needs nutrient cycling, which means that once the plant is making glucose, then the saprotrophs is crucial in order to continuously break down the molecules in order to be used by the soil again and again. So the recycling of waste also is important in order to make this sustainability possible. And here is the link of this guy, mesocosm, that his mesocosm was survived for 50 years, and I have no idea if it is still alive. This guy is alive or his mesocosm, but this is the famous picture around the Google if you do it. Here is how to make a mesocosm video. You can watch the video by yourself. And here is the blog of how to make a miso custom. Miso custom is one of our activity, and you need to do it and prepare it and keep it and keep it in the lab for the next year to see how long will be survived. And that's the video. Yeah, thanks for watching.